Studying the complex genetics behind hair color reveals how melanin affects us. One of the traits that we usually use to physically describe people is their hair color. Hair is a useful descriptor because it varies so much among us. Melanin is the molecule responsible for the many different hair color tones. It's also responsible for the color of our skin and eyes. We inherit these traits from our parents in a complex way. Understanding how our genetic information can produce different hair color tones can be as difficult as entangling long hair after not brushing it for several days. Even though some genes are known to determine hair color variation, recent studies based on large cohorts of people from the United Kingdom and Latin America have shown that there are more than a dozen genes involved in hair color. In a recent paper published in Communications Biology, my colleagues and I studied the genes involved in hair color in a Canadian cohort of nearly 13,000 individuals of European-related ancestry. Our findings provide insights about genetic variants that may be driving differences in hair color. Types of melanin. Melanin is produced in a specific cell type called melanocytes found in skin, eyes, and hair follicles. Melanin is also found in the brain. The type and amount of melanin and how it is distributed in cells is what creates differences in hair, skin, and eye color. Melanocytes produce melanin, which in turn affects skin and hair color. Melanoma, a kind of skin cancer, consists of an overgrowth of melanocytes in the skin. There are two main types of melanin in our hair, eumelanin and pheomelanin. Eumelanin is also known as the brown-black pigment, whereas pheomelanin is known as the red-orange pigment. People with red hair have much more pheomelanin, people with dark hair have higher levels of eumelanin than pheomelanin, and blonde hair is due to low amounts of both pigments. The main difference that guides which of the two types of melanin is synthesized is a switch in a protein called the melanocyte-stimulating hormone receptor, or MC1R. Variants of the gene MC1R that lead to a loss of function of the protein can affect the production of pheomelanin. In contrast, there are many genes across our genome involved eumelanin variation, including less damaging genetic variants in the same MC1R gene. Entangling genetic complexity. In our study, we used genome-wide association studies, GWAS, to identify genetic regions associated with hair color across our autosomal chromosomes. GWAS identifies overlapping associations in a gene of interest or other functional genomic elements. This method also identifies associations in intergenic regions, the DNA sequences located between genes. Correlation does not imply causation. Therefore, we also worked on obtaining evidence to locate genetic variants within or near genes of interest that are more likely to be causing hair color variation. This helps us better understand the molecular mechanisms involved in pigmentation. We identified genetic variants that have been previously reported, such as those damaging the function of MC1R, which can result in less eumelanin production or even switch the production to pheomelanin. Other genetic variants identified that are involved in hair color and pigmentation in general do not change the protein's structure or function. Instead, they regulate the expression of the gene, which means that they control how much of a protein is produced. Vitiligo is a genetic condition that affects the production of melanin. One example is a genetic variant near the gene OCA2, in which the gene expression of OCA2 decreases in the presence of guanine, one of the building blocks of DNA. This results in lower melanin production. We also tested if the associated regions shared genetic signals with DNA methylation, which can regulate gene expression, in melanocytes. We observed that the DNA methylation state may be a relevant process in regulating pigmentation in some genomic regions. Future investigation is needed to provide concrete evidence on this. Pigmentation pathways. By studying the genetic factors determining hair color, we can increase our understanding of how pigmentation occurs. This helps us further understand pigmentation diseases and their genetic risk factors, such as the role of pigmentation in cutaneous melanoma and in vitiligo. Another interesting application is the improvement of prediction models of hair color from a DNA source, which has implications for forensic DNA phenotyping in police investigations, which predicts what someone looks like from forensic samples. Including other population groups in the research of hair color may help us identify new genes, which can further improve our understanding of pigmentation mechanisms. Frida Lona Durazo, Postdoctoral Fellow, Computational Genetics, Université de Montréal.